Welcome everybody, welcome to the session that we've named the session Bringing a Transformational Technology to Market, the PRRS-resistant thing. And the source of this session actually started last year when we had one of the keynotes to start talking about gene editing and PRRS-resistant pigs. And back then, last year, we actually had a lot of questions that stayed unanswered out of that session, out of that keynote. So we just intended to, in some way, carry on that conversation to this year, and that's what we are trying to do in this breakout today. So we are gonna, actually, that was the title of your talk, what I just read. So sorry. But anyways, you know what we're going to be talking about. So to do that, we have invited Dr. Lucina Galina. Dr. Galina obtained her DVM from the UAM University in Mexico City, and then she completed a PhD in swine medicine under the mentorship of Dr. Carlos Pijuan at the University of Minnesota, worked on PRRS actually back then, and Streptococcus suis. She had a stay in Europe, in the UK, also in a research center, and then she joined PIC USA for over a decade, where she worked on the area of genetic markers for disease resistance and robustness. And later on, Zoetis led the team at Zoetis, a team of veterinary professionals as director of the Swine Technical Services Group. And more recently, she's back at PIC as Director of Technical Research, and the project that she's responsible for has to do with PRRS resistance. So Lucina, if you would like to take it from here, thank you.
Okay, good morning everyone. It's a pleasure to be here at the University of Minnesota, my alma mater. So thanks for the invitation, Monse. Monse mentioned that I worked for PIC from 1998 to 2009. 11 years of my life, and what we were looking for was we wanted to find gene markers for disease resistance. So we did a lot of studies with Salmonella, Cholera suis, with Haemophilus parasuis, and then later on with PRRS. And after all of those 11 years, what we found, we only were able to find differences in susceptibility. Animals were more or less susceptible to these diseases, but we were not able to find true resistance. What I'm going to talk to you about today is radically different from those findings. We're talking about resistance to PRRS. So it's using gene editing. We are going to be talking about a transformational technology. So I was here, as Monse said, in the early 90s when the first virus was actually being discovered. And the University of Minnesota took an important role in the discovery of the virus. And I'm sure you are familiar with this slide from the Swine Health Monitoring Program. In spite of 30 years later of all the investments in technologies, we filtrate farms, we vaccinate them, we move them, we have enhanced the biosecurity, we move animals in certain ways, PRRS continues to be a big challenge. And you can see that today. We're having this PRRS session here, and there's another simultaneous presentation also talking about PRRS. So we're not out of the woods, unfortunately, and in this Swine Health Monitoring Program report from September, this graph represents 50% of the sows in the US. And if you just focus, you have information from 2009 to 2023. And if you just look at the green and the red, it's like a traffic light system. Green is good, so these are farms that are negative, and red is bad, these are farms that are positive. What we see is an increase over time of positive farms, sow farms, and a decrease of positive farms. So in spite of all of these efforts, sometimes it feels that we're going kind of backwards. This is a picture of David McGee. He's one of the colleagues at PIC. And that picture is the first pig that was born from a gene-edited parent. So the very first one, this picture is about 6 years ago, almost to this date. And the parent, one of the parents of this pig was an intentionally gene-altered, a parent that had the gene in question, and the other parent was a regular parent. So this is basically what we call generation 1 or E1 generation. After that, the next generation of pigs is called E2 and then E3. What is important for you to understand is that the parent of this pig, one of the parents, had the edit. But then this trait is inherited through Mendelian inheritance. So doesn't mean that this pig is genetic. It was created, or it was originated from an original founder parent that has the edit. Sometimes people think this is not a reality yet, that is a kind of a technology that is not here. You know, tomorrow I'm going to be heading to one of our farms. We have a few farms here in the US that have these pigs. We don't have an approval yet from the FDA, and we'll talk about that. But we have animals that are alive, that look like pigs, behave like pigs, that are just normal pigs that are generated today. So I'm going to give you a brief introduction to the technology, and I think it's important as veterinarians that we understand how the technology works, because if your customers ask you questions, that you're able to relate to this. But probably you recognize the faces of these two beautiful ladies, Jennifer Doudna and Emmanuelle Charpentier. These two ladies work with Streptococcus pyogenes, and in 2020 they won a Nobel Prize because they discover or put together information about this technology called CRISPR-Cas, which is basically what we call gene editing scissors. So these two ladies did some of this phenomenal work, and 2020 is not too long ago. So all of this is recent and is moving really, really fast. But basically, when you hear the words, you will hear gene editing and you will hear CRISPR and Cas. So when we talk about CRISPR, we're talking about clustered, regularly interspaced, short palindromic repeats. It's a mouthful, and you don't have to memorize all of this. But these genomic tools are a part of the DNA of, it was discovered in bacteria. And what they are basically here, so we have this genome, this is a bacterial genome like Streptococcus pyogenes, and what this is repeated sequences, short sequences of DNA that repeat themselves, and then they're interspaced by these blocks of DNA that nobody knew what they were. And then more repeated sequences, and another block of a different type of sequence that nobody knew what it was, and then more repeats. These repeats are short. They are just a few nucleotides, maybe 20 or 40, and they are palindromic. So if you unwind them, it reads one way, the nucleotide sequence, it reads the same way, with one of the sections of the DNA than the other. For example, if you look at the word Hannah, you read it one way, it reads Hannah, and then if you read it backwards, it's exactly the same way. And there's a reason for that. But it was curious that they had all of these repeated sequences and people didn't understand what they were. At the beginning of the CRISPR-Cas complex, there's a group of genes that are called CRISPR-associated proteins. And these Cas genes, CRISPR-associated proteins, have the role of unwind DNA. So you see a zipper, it unwinds DNA of a target DNA, and then it cuts different portions of the DNA. So this is the tool that is going to allow you to get to specific sections of DNA, any section of DNA, and target an area that you want to cut. Now let's go back to the mystery boxes. So they knew what this was, the repeated sequences, but they didn't know what this was. And the answer came again from bacteria. So the arch nemesis of bacteria are viruses, like in our lives. And these are called bacteriophages. And what the CRISPR-Cas system, so here you have Cas, and then you have your repetitive DNA pieces, and these clusters of unknown DNA, it turned out that the sequences in here, and there should be another here and another here, were were pieces of viral DNA that was found in the bacteria. And not only that, it was viral sequences that attack Streptococcus pyogenes or other bacteria. In essence, the Cas and CRISPR complex is kind of a library or an immune system for the bacteria to be able to recognize viruses that infect the bacteria. So you have a new a virus, maybe a virus that this bacteria was exposed to before. It injects the genetic material. So the Cas CRISPR complex is going to recognize, oh, I've seen this virus before. And then using these sequences is going to help, and the use of Cas is going to help locate the sequence in the target sequence, and using the Cas proteins is going to be able to snip and cut this foreign virus. So it's a way to protect themselves. So what Jennifer Doudna and Emmanuel Charpentier did is, like, they said, hey, can we harness this technology and use it in a way that we can determine what we want to cut? And that's exactly what we did. What they did, they are using Cas9, which is one of the easier proteins to use, and they kind of simplify the process so that it's friendly to use in the laboratory. So this was a breakthrough technology, because with this now we're using this technology to target cancer, targets. It can be used to target components that can help with sickle cell anemia. And in our case, it's exactly the technology that was used to target the receptor of the PRRS virus. So this was a breakthrough technology that allowed us to come to the next step. Let's talk about the PRRS gene edit. So probably you are familiar, there was a lot of discussion about what is the receptor for PRRS. I think we are past that. People understand that the receptor is CD163, and there's a talk that is happening exactly at the same time, and Jay Calvert from the Zoetis or before Pfizer, he's the one that discovered that if you have a cell that is not permissible to PRRS, and you add this receptor by genomic means, you make this cell permissible. So we know that this receptor is very important. And if you imagine this is the cell, and remember for the target for PRRS is the monocyte and the alveolar macrophage, is the only target for PRRS. And this is, imagine that this is a monocyte and this is the CD163. The CD163 is like a pearl, like a necklace of pearls. And there's 9 pearls, 1, 2, 3, 4, 5, 6, 7, 8, 9. And we now know that it's actually pearl number 5, which is here. Sometimes we call it domain 5. This is precisely the place where the virus attaches. So at the beginning, the University of Missouri, the group of Dr. Prater there, first they created a gene edit, eliminating the entire CD163 gene. And that confers resistance to PRRS, because then the virus doesn't have anywhere to attach. But then later on, the Roslin Institute only created an edit that just removes this domain, because genes have functions, and there are other functions that are important for this gene. So the edit that we're using at PIC targets only this domain 5. And then what happens is that when you remove that domain using Cas9 and CRISPR technology, then you don't have this domain 5, and the virus is unable to attach. It's there, but there's nothing to attach. So it cannot infect the macrophage, and therefore it cannot infect the pigs and cause disease. So that's basically the understanding of how things work. So let's just briefly talk about the PRRS receptor. I think it was one of the questions that was hanging on from last time. PRRS genes have functions, and CD163 also has a function, and this is just a copy of a paper that describes the functions here. The main function of the CD163 is to remove hemoglobin from the system. It's a basic physiological function of, for organisms, not only pigs, but many organisms. If you have a lot of hemoglobin circulating, it can damage the kidneys. So these complexes remove the circulating hemoglobin. So it's called the haptoglobin hemoglobin complex. Other functions, and these are in different domains, different than the domain 5. Other functions is it helps the formation or the maturation of red blood cells from erythroblasts to erythrocytes. So that's another important function. A TNF-like receptor, this is one of the receptors, that's another function. a receptor for some bacteria. And then here we have viruses. You might find in the literature that CD163 has also a role in the attachment of African swine fever. And this is an area that we're investigating because it wouldn't it be nice that we also have a marker for African swine fever. But it turns out that when you remove the receptor, the virus, the ASF, still can invade the cell. So it uses this gene, but it also uses other receptors. So African swine fever, they don't work for this setting. But obviously, what I'm talking about now is that we now know that this domain 5 is also the receptor for the PRRS virus. This is as far as we know about other pathogens. People often ask, hey, are there other pathogens? Is this pig resistant to other pathogens? As far as we know, PRRS is the only one. And another important thing, if you're wondering, wow, you're going to remove genes or pieces of genes, pieces of proteins from an important receptor, no specific role has been associated with domain 5 other than PRRS attachment up to this point. I would like to make an important distinction because people, the general public, don't really understand this distinction, and I think that we, as veterinarians, have a role in explaining the difference. The difference is what is a PRRS gene edit and what is a GMO. And sometimes people say, yeah, watch out with this PRRS, it's a GMO. It is not. So the PRRS gene edit, you have a portion of a protein. So you have here, let's say that this is a protein and these are different portions. A portion of the protein, like domain 5, is removed. So it's a tiny piece of sequence that is removed, but no foreign material from any other organism is inserted. In the case of PRRS, the PRRS gene edit is a removal or a deletion. This is different from a GMO because with a GMO, what often happens is that you have a piece of genetic material from a different species, like in the case of the corn that has genes from a different species, like from a bacteria, that is inserted into their genome. So that's a very clear distinction between gene edit, a removal of a receptor or a portion of a gene, versus the insertion of foreign material. So very quickly, the path to commercialization here, we are awaiting for FDA approval. So the regulatory agency that allows us or not to use this edit is the FDA in the US. And we have now submitted all the paperwork. There's this very stringent process where we have to provide a lot of information to show that it is replicable, that it lasts through generations, that a pig is a pig, a number of packages we call them. The submission has taken place. So now for us, the clock is ticking. We have 180 days to wait for the answer. So our hope is that by the end of Q1, we are getting a response from the FDA. Here we have other 10 kind of strategic targets that we're focusing on first. And after the US, you can see that there are, you heard Randy Sprung talking about how complex is our, the pork distribution or export system. So in the US, we export 30% of pork that goes to Canada, Mexico, Japan, South Korea. So we're looking for approval in these countries. Other countries like Colombia, they say, well, this is not a GMO. To me, this is a pig, normal pig, so you don't have to have a regulatory process. We consider these pigs acceptable pigs to bring to this country. So here are our targets, and the US, we are making advances, and other countries like Colombia, they don't have those requirements. Other countries like Brazil, you must be wondering why they're in the list. And you heard Randy talking about how important this market is, for pork production, and they are PRRS negative. But perhaps Brazil wants to protect the market. Perhaps they don't want to become positive in the future. So that's an important target for us to have in the list. So what happens if we get the approval at the end of Q1 in 2024? So there's a few things that are important for you to remember or understand about this genetics. PRRS resistance is transferred just through Mendelian inheritance. But what you need to understand is that this is a recessive gene. Resistance requires actually two homozygous resistant genes. So imagine that you have a heterozygous father and a heterozygous mother, where the lower A is the resistant gene. The 50% of the population will be heterozygous. That means that they are not going to be resistant. 25% of the population is going to be null. They don't carry on any of the good genes. They are not going to be resistant. 25%, this is the blue pig, is the only pig because it carries two genes that are resistant. So that's important to understand because once we get an approval, it's just not going to happen overnight that you're going to have all of these pigs ready to go. The most likely scenario for implementation is delivery to customers using semen. So via semen is the most likely scenario. But implementation takes time, and that's also important to know. People think sometimes that it's like a light switch, and it is not. Here's an example of 1,000 sow herd where we're introducing, this is a hypothetical scenario, introducing resistance through the edited semen. This system produces about 23,000 market pigs. And as you can see, just by introducing semen and growing year by year, by year 5, you would have 70% of your population resistant. You can accelerate this. Perhaps you can introduce gills that are carriers of the gene, or you can increase your replacement rate to accelerate all this process. So there are other ways that geneticists can deal with shortening this time. But it's not a light switch, but it's more like a dimmer that is going to go a little slower than that. All right, so since I rejoined PIC in February, I have been conducting a lot of studies to see what does the PRRS resistant look like in vivo. And this is what I wanted to share with you today, perhaps the most important piece of information. I'm going to describe the results of basic experimental studies in nursery pigs. So our typical study, we have lineage pigs that are homozygous resistant, null, or heterozygous. We acclimate them for 1 week. We use Midwest Veterinary Services in Oakland, Nebraska, because they have FDA, USDA, quality, the way they conduct their research is approved by these organizations. And then we challenge them intranasally because we want to use the natural route of inoculation, and then we follow them for 3 weeks. And then we do PCR analysis to understand if the virus is there and if the immune system is recognizing them. So this is one example. This is a 1441C strain, one of the pathogenic strains that is circulating in Minnesota, in Iowa. And here we have about 12, 13 pigs that are homozygous, so they have 2 copies of the resistant gene. And then we have a mix of heterozygous and null pigs that they shouldn't express resistance. All of them come a negative herd, so by day 0, before challenge, all of them are negative by PCR, and they are negative by negative by PCR, all of them here, and obviously they should be negative by ELISA. Once we challenge them at day 3, you can see that all these homozygous pigs, we cannot detect the virus via using PCR from blood. We just don't detect that. And if you look at serology, you would expect by day 10, you should be seeing this seroconversion, and we see no seroconversion. All of these animals are housed in the same place. They are challenged all together, and in the environment, the virus is there. We just simply we cannot detect the virus. And this is obviously very different in the case of the heterozygous and the null pigs, which get infected by day 3, and as you would expect in any normal animal, they do seroconvert. So these are the results of one pretty nasty strain, the 1441C. In the past several months, we have conducted more research to kind of increase our portfolio of strains. You know that if you look at the prevalence of PRRS in the US, 41% of the strains coming to diagnostic labs, whether it's Minnesota or Iowa, are line 1Cs, 24s are line 1H, 14 lineage 1A, I'm sorry, it's lineage, not line. So lineage 1 dominates, 90% of the strains that are circulating today are lineage 1. 8% are other lineages, and then 2% are other, like PRRS type 1. So I here simplify, this is the study that I showed you just now, but our results with lineage 1H and the strain 1A4 is kind of the flagship representative strain from this group. We see exactly the same results. Homozygous or edited pigs, just by PCR, we just don't see the virus, while the control pigs, the virus is there just like you saw in 411. Lineage 1A, 174, you guys have had issues with farms with 174, exactly the same results, the same results with 142, and then we are using other lineages as well. Sometimes if the strain is not very pathogenic, we see that they become PCR a little later, or in the case of this is a PRRS type 1 strain, it takes a little while, but then eventually all of them get infected. So we have now repeated this work multiple times, and we can see exactly the same results. Another question that Monse posed is, what happens? What do these pigs look like? Are they exactly like a non-edited pig? So in a nutshell, because I'm running out of time and I got the signal from Monse, as part of the regulatory package, we have to demonstrate that a pig is a pig with edit or no edit. So we have to include information from early life. We have edits in 2 male lines and 2 female lines. So for regulatory purposes, we include data from a male line and a female line, and we look at birth weights, defects, cryptorchidism, hernias, mortality, finishing traits like lifetime daily gain, carcass weight, mortality, reproductive data. And if you see unequal is basically, and we run a statistical analysis, they are exactly the same. When you compare a homozygous pig versus a heterozygous agno pig, the pigs behave exactly the same. Other traits, carcass and meat quality, you can see the information there, and composition, amino acids, proteins, all of them are behaving exactly the same. So, in a nutshell, homozygous pigs are expected to control the effects of PRRS, improving clinical signs, improving, reducing mortality. I cannot think of any tool that can improve the welfare of a pig than having a pig that is resistant to PRRS. We think that gene edit offers an unprecedented opportunity, obviously for PRRS, which is the biggest problem that we deal with in the US and in other parts of the world, but it is also can be viewed as a platform for other solutions different than PRRS. We are focused on enabling a successful commercialization of the PRRS-resistant pig, and for us, having market acceptance. So what the public think, not what the farmer or the veterinarians think, but very importantly what the consumer think is very important, because we know that without that acceptance, we're not going to be successful. And our next speakers are going to tackle that issue about acceptance, especially at the end of the pork supply chain. So I'm going to stop at this point, and I think we may have a few minutes for questions.
Thank you very much, Lucina. I appreciate the presentation. We do have time for questions, and for the ones standing here, if you would like to take a seat, feel free to do that. But we do have time for questions. So, Lucina, one question that keeps coming up is whether how do these pigs perform in the face of other challenges, like growing in a finishing site in Iowa or Minnesota that has plenty of influenza and plenty of E. coli. How do those pigs perform?
So right now, we have a lot of restrictions on where to have those pigs. We need a special FDA permission. So we have pigs, the pigs that you saw in these challenge studies are grown in pristine conditions, and they are sent to a contract research organization for challenge. Obviously, once we get the approval from the FDA, we want to bring those pigs to those conditions, like a more real life, where maybe you have 2 or 3 strains of PRRS coming, maybe you have influenza. And we might be able to do that through our gene crossbred program, in the future. But so far, those pigs cannot be slaughtered. They have to be euthanized. They cannot leave the confine of the facilities. But obviously, that is a good question for us, and we want to bring those pigs to a more challenging environment in the near future.
one question there, and then.
Thank you very much. What's the function of domain 5? It's just there so that the virus can attack, or does it have another function?
There is no known function for domain 5 so far, and that's very important for us to understand. We know that the other domains have specific functions, which I listed there, but there has not been any report of another function for domain 5. And that's why it's so important for us to check other traits, to check the milk quality.
Does the CD163 has to do with blood work? Did you do all the blood work?
Yes, some of the other domains, yes. But there's no known function at this point for domain 5. Yes.
And Camille, one question.
I don't know if I'm right, but I read somewhere that the USDA will call that technology IGA, International Gene Alteration.
Intentional gene alterations, that's right.
How the public will receive that, and are they going to make the difference between IGA and GMO and all of that?
Well, that is a key question, and I just came from giving a series of presentations in the UK, and that's the question that people ask. Lucina, you just explained the difference between a gene edit and a GMO, but the public may not be able to understand that. So I think I'm going to let that one for one of the next speakers, because David Fikes is coming from this institute that lobbies for all the retailers. He is at the end, and this is not the first time that he's dealing with this technology. So let's hold that, Camille, and give us some time to David to give us his thought from the customer perspective, not my perspective, on this question.
Thank you very much, Lucina. Appreciate the presentation. Thank you.